The beetle genus Neoserica is a polyphyletic mix. It's a catch-all category where highly uniform dorsal carapaces obscure actual evolutionary history. Historically, species delimitation relied heavily on these outward appearances. Because so many of these beetles share nearly identical coloration, researchers mistakenly lumped entirely separate lineages into single taxonomic categories. Overcoming these historical errors requires shifting from macromorphology to micromorphology. To find true species boundaries, taxonomists must dissect specimens and examine their specialized reproductive structures. The core diagnostic feature is the male Aedeagus. Specifically, researchers map the precise structural geometry of the asymmetrical left and right parameres. This specific taxonomic revision has two operational objectives: resolving historical synonymies and describing newly discovered cryptic species that were previously indistinguishable. In clades like Neoserica, biodiversity is not a matter of what is visible on the surface. It is encoded in the exact structural mechanics of reproduction. Before reaching the parameres, we filter the Saraceni tribe down to the correct genus. This requires identifying two physical markers: a carinate hypomeron on the underside and a bidentate protibia on the front legs. From there, we isolate the Neoserica lubrica group. These beetles are defined by a small body size of 6 to 8 millimeters, a yellowish-brown hue, and a completely shiny, glabrous dorsal surface. This contrasts with the Neoserica obscura group, which presents a completely dull exterior and is typically bicolored with black and reddish-brown sections. Both groups do share a moderately sinuate apex on their metatibia, which keeps them closely adjacent in the taxonomic key. While these macro-diagnostic traits successfully separate the broad groups, they are unable to differentiate the individual species within them. External traits serve only as a geolocator to reach the correct clade. Actual species identification requires coordinate-level precision on the parameres. These habitus photos prove extreme visual homogeneity. To tell them apart, we extract the Aedeagus. Neoserica fugongensis establishes a baseline left paramere curvature, moderately curved, bending strictly externally at the apex. In contrast, the identical-looking mantillary reveals a blunt central angle, bending sharply backwards. Angles aren't the only diagnostic markers. Ratios are just as critical, which brings us to Neoserica dongjaffinensis. Isolating the left paramere of dongjaffinensis, we measure the distal hook against its total length. The hook is significantly shorter, comprising less than 1/4 of the entire paramere. For secondary conformation, the basal portion is nearly as wide as long and distinctly lobiform. This specimen photo shows the singular outlier in this clade, Neoserica menglunensis. It can be identified without dissection by examining the labrum, which features a transverse rim of dense seta, a structure absent in all other Chinese members of the lubrica group. Structural mapping corrects decades of historical literature by identifying where separate lineages were mistakenly merged within the obscura group. These technical diagrams of the Neoserica obscura holotype detailed the specific structural identity of its parameres. This exact geometry acts as an anatomical fingerprint. When researchers dissected two other supposedly distinct species, Microserica rohri and Maladera chinensis, they found their parameres were identical to obscura. For years, minor geographic variations were mistaken for true biological divergence. Cross-examining the type specimens proved they were all the same organism. But while those names were collapsed into synonymy, examining these specimens revealed a legitimate cryptic look-alike in the same clade, the newly described Neoserica allobscura. To separate it from the true obscura, we have to examine the curvature of its right parameter. Notice the strong ventral bend right in the middle of the right parameter structure, a feature completely absent in the true obscura. For secondary conformation on the left parameter, its dorsal lobe is shifted much further basally, and its tip bends inward. Without identifying these specific microstructural signatures, the discovery of allobscura as a distinct biological entity would remain impossible. Systematic revisions ensure that our species lists reflect biological reality rather than human misinterpretation. Finding order in the Saraceni tribe requires a strict sequence of filters. We identify the carinate hypomeron to isolate the genus, and then rely entirely on parameter geometry to identify the species. This revision of Neoserica proves that biodiversity cannot be accurately quantified by external phenotype alone. Relying on precise anatomical data remaps our understanding of how these organisms are distributed across southern China and Indochina. This morphological signature map shows how the structural architecture of the male Aedeagus is the final word in defining a species. Taxonomic papers like this are essential for resolving polyphyletic mixes and bringing objective rigor to evolutionary biology. While the surface of nature is often conservative, the underlying mechanics of reproduction remain the ultimate arbiter of evolutionary truth.